directly. So yeah, Armita, the floor is yours.
Yeah, thanks, Hanflo. I'd like to thank first all the organizers for this very exciting meeting. It's really good to see familiar faces after all this time. Um, the story I'm going to talk about today is sort of a topic we've started working on control of molecular evolution. Maybe the title is a bit overhyped, but we started sort of learning whether or not we can control evolution and how to think about this problem. So I want to share some of these insights with you today. Uh, so the idea of controlling evolution is not really new. So the first time this was brought up was basically by Wallace in a letter that he wrote to Darwin about the fact that natural selection, as they were calling it back then, or evolution in general, should be thought as basically a centrifugal of governor of the steam engine, and it works as a control machine to checks and corrects basically irregularities in species by compensation. So he was thinking at the time that evolution, which was basically expressed as tendency of varieties to depart indefinitely from the original type. They didn't have a name for it. So that's basically how a control works. A control in a machine works. So these things are some sort of balancing each other in a controlled sense. So this is not the way I want to talk about control, because we know, well, we have some ideas how to think about evolution these days, and maybe putting a strategy on it is not the best of ideas. But what I'm sort of interested in is thinking about evolution in terms of how do we intervene with natural selection to drive it in a direction that that is desired. So human population have been doing that for a very long time in the context of selected breeding. You can see here basically a Novosibirsk fox populations that they were breeded to be domesticated, and you see, the floppy ears and some dog type behavior of these foxes that emerged in Russia back then. We all are familiar with how the high yield tomatoes that taste really bad and taste basically like nothing. So this is a result of a selected breeding that didn't really take care of the taste much, was just a look. So this is a topic we've been looking at in the context of phenotypic or multivariate phenotypic control for directed evolution. And it's something I will talk about sort of on the theory side today. Another sort of control approach that my lab is working on is thinking more in the directed evolution and high throughput experimental directed evolution setups. So this is a collaboration with the group of Yannick Wunderles and run by Vincent Ballardi in Paris. And in this context, what we are really interested in is molecular design and optimization, in particular, optimizing function of a polymerase and trying to understand how various factors impact evolution of polymerase in the lab in an in vitro setup, in sort of a microfluidic experimental setup for directed evolution. And the last thing that sort of we are working on these days in the context of control is a therapy design. And in this sense, what we are interested in is how to use various sort of elements of control, sort of therapy control, including antibodies and drugs to curb viral evolution, in particular HIV evolution within patients. So I will talk about two of these things today. The first one sort of more on the theory side, and the last one on the therapy design to show you some data and some of the analysis that Colin Lamont has been doing in the past year. To start with, let's start with the theory. So this is a work in collaboration with Jae-Hyun Exin, who is now at Texas A&M, and hopefully it'll show up soon in Physical Review X. So the idea here was a simple one. Let's imagine you have a population of cows, sort of a bunch of pink cows in a population, and the usual white cows that are sort of reproducing and are under positive selection, so they sort of reproduce better, have a higher yield under natural selection. But the desire of the farmer is to have more of the pink cows, but without compromising with the yield. So how do we sort of control this population and selectively breed not to compromise for these multivariate phenotypes? And the key here is to observe a population, implement an artificial selection protocol, and then have a feedback on your natural selection on the natural phase of evolution, and gradually drive the population towards a desired target. And it may sound, the objective function is clear, but how to get there is can be complicated. And that's what I'm gonna talk about today. So the ideas of controlling these kind of evolutionary processes have been around for a while. And so more recently, we've seen experiments in the bacterial sort of experimental evolution of microbes that implemented control protocols for that. So one example of it is morbitostat that was designed by Erdal Toprak in 2013. So in that case, basically the desire was to keep bacterial evolution evolving and adapting. So for that, you want to increase the selection pressure or the amount of antibiotics that you have in your culture for the sort of resistant mutations to keep popping up in the population, and the population won't sort of flatten, the growth rate won't flatten. So what it does here is like they use some optical detection system to measure the growth rate, and they tune the amount of antibiotic in the culture based on the growth rate of the population. So that's a sort of observable, and how they implement the feedback in some sort of a control, well, what you would call a bang control setup. A more recent approach, which basically came out last year from the lab of Chang Liu and Ahmad Khalid, was sort of a continuous, high throughput continuous evolution, or automated continuous evolution, they call it ACE. So in this case, they have set up a system that uses a coupled yeast polymerase and plasmid that you can implement targeted mutagenesis in a high throughput way in there. And they also implemented an algorithm, sort of attached an algorithm to the system, a control algorithm of PID, which is basically a sort of control algorithm you're all familiar with as the first control you learn, which is a cruise control algorithm, which is called proportional integral derivative control. They implemented this algorithm in the experimental setup to drive the evolution towards desired targets, and they're using this for improving enzymatic activity, some protein functions, so a sort of a molecular level directed evolution. So it's pretty effective because we know PID. If you have used cruise control, which most of you have probably, you have a very fast response time, and it limits the error that accumulates over time. So by integrating over the history of the trajectories. So in a sense, it's a rather effective approach. The problem with this PID approaches is that you need to tune a lot of hyperparameters in your system. And when you design a car, BMW knows how they design the car and how they have to implement cruise control. Whereas in an evolving population, it's an experimental setup, and it might be complicated to do that. So what I want to talk about is how do we think about sort of controlling these systems, maybe in a simpler toy models, without having to fine-tune a lot of parameters. So the goal is the following. We want to come up with optimal control, perhaps in a finite horizon, within a finite amount of time, or in a steady state. We want to observe the system, implement some optimal intervention. We might have to do this control not continuously over time, but rather with some scheduled implementation. We want to maybe optimize this scheduling based on the value of information we gather from these evolutionary trajectories. And we want to learn sort of the, in the sort of most complicated case and maybe most realistic case, we want to both learn the evolutionary parameters and implement the control at the same time. So this would be for the cases that we don't have actually a good model of evolution, which is basically in most cases. So that's a sort of high level idea of what the problem is in a theoretical way. So that can be control for phenotypes, control for mutant frequency, or anything you wish for. The problem is subject to noise. We know stochasticity plays a big role in evolution, and in sort of optimal control problems, stochastic effects are important. When you think about engineering systems, there's an example of called drunken spider. So if you were to, if you have a spider that wants to sort of cross a river, go to the other side, in normal days, the spider would take a shortest path, go on a bridge, and get to this house on the other side. But if the spider is drunk, meaning its trajectory would be more stochastic, crossing a river actually has quite a bit of risk, because there's a good chance the spider falls in the river. So you have to, well, a lake. So the spider needs to go around this lake. And so that might be sort of a longer trajectory, less optimal, but if you have a stochastic dynamics, it might be more beneficial. So that's basically the underlying rules that we need to observe. And we will try to sort of think about simple stochastic processes that that underlie our evolutionary dynamics to try and implement control protocols in our setup. So the model that I'll talk about today is evolution of covariant phenotypes. So in this case, we're using the simplest way of thinking about it, and that would be having a multivariate quadratic fitness landscape. So the vector X is a phenotypic vector, in principle a k-dimensional vector, and you have a curvature matrix C that basically gives you a strength of selection in this high dimensional space. In principle, so this is projection in 2 dimension, to just look at it, if you have a natural selection, yeah, so we set also the 0 as the point of optimal fitness. So if you run your population by usual sort of Wright-Fisher process, you have an element of selection that couples to the diversity in your population. So K here is the covariance matrix of your phenotypes, and derivative F is the gradient of your fitness function. So that's a selection component. You can have equivalent mutational field that couples to, again, a diversity in the same way. And you have a genetic drift, which is basically proportional to your diversity. The strength of genetic drift would be proportional to your diversity and inversely proportional to the effective population size, whatever that means in this context. So if you run your population, as we all expect, it will sort of move around the optimal fitness point in this high dimensional space. But if my desire is to drive a population away from this 0,0 optimum or natural optimum towards an X value that is, in this case, 1, and keep the Y value still very close to its natural optimum, then I need to implement a control protocol, which I call it UX of T. And it's, it's a state and time dependent protocol, depending on whether or not I have a finite horizon for my control. And so if I implement an optimal protocol, I would be able to drive my population within a desired point time towards a desired target. So how do we compute this? So to be specific, we have an objective function. This objective function is we want our phenotype x at time t, for example, to be close to some target x star, and we have some cost matrix G, so which would measure the deviation from this target. And we want probably to minimize the amount of amount of control we want to put in the system. So you don't want to really hammer the system. So you also want to minimize this cost of control. So the amount of U you put in. So the goal is to minimize this omega function. And you can also have what's called the endpoint cost, which may be different than the path cost of this thing. So you integrate over the omega function over time from the initial time point to the final time point of your interest, plus a endpoint cost if you wish. You want to minimize, you want to find a U function that minimizes expectation of this sort of objective function over evolutionary trajectories that you have. And there is a standard way of sort of formulating this problem, and it's called Hamilton-Jacobi-Bellman equation. And you write an equation for this costical function and try to solve it. In general, it's a complicated problem. It ends up being a sort of nonlinear PDE to solve and not always easily solvable. But sort of numerically, you can solve it a lot of times, and it basically comes down to a way of thinking about this problem in backward in time or dynamic programming, and that's where name of Bellman comes. Most of you know Bellman from dynamic programming. The idea is to sort of start off from target and try to find an optimal trajectory that takes you to the starting point. There are a class of problems that we can solve that are easier to solve in this context. And that's how we formulated our problem. It's called sort of path integral control problem, in which there's a relationship between your cost function and the stochasticity in your system. And so the key here is you want to control the more variable degrees of freedom more than the sort of strict degrees of freedom. And it's somehow intuitive because when something is very conserved, there's a good reason probably for it to be conserved. You don't want to really sort of perturb that degree of freedom much. So if you sort of assume that, you can write down the control problem in a sort of easy forward way, which helps solving these equations. And you do some math, what you end up producing is the sort of evolution equation for your probability under control, which I call it probability of trajectories under control, which I have indexed U for it, which would be sort of your stochastic dynamics under natural selection, so your uncontrolled dynamics, minus an annihilation term, which basically kills off your trajectories with a rate that is proportional to deviation from this target, which was this V function we introduced in the objective function in the previous slide. So what it does here is that if you start on a trajectory from one point, and my target here is at value 1, depending on deviation from this target, I basically kill off my trajectories at different points in time. And as you do this, as you implement this algorithm, you end up producing the resulting point, the sort of ensemble of survived trajectories end up looking like a optimally controlled sort of state in the system. So this would be probably, if I run the simulation in this case, I would generate 2 ensembles. So this is the natural ensemble under natural selection, and the blue one would be under artificial selection. That's just by killing off trajectory. So that's a easy algorithm to implement. But also, assuming this sort of regime of evolution, you can solve the problem in some cases analytically, and that's what we did for the case of multivariate phenotypic evolution. So to show you some of the results, one thing that we managed to show is that if I try to basically drive an evolution towards a target in one phenotype and keep it close to its natural selection on the other phenotype in this case, I end up moving my fitness landscape and designing an effective fitness landscape and moving its peak towards the target as time gets closer to my target time. So the colors here show relative time to the end point. The different shapes here show the phenotypic correlation, so between X and Y that is in your system. So if your phenotypes don't co-vary with each other, then this is basically the line you need to go when your phenotype, you don't change the fitness function on your phenotype Y because you want to keep it close to its natural point, and you just move the phenotype X. If they co-vary, you need to counter that. And as the relative cost of controlling Y to X changes, so we go to the right, is a cost of X is higher than Y. What you end up is you leverage the less costly phenotype to move the more costly one. And that's what is happening here. So I basically end up first moving the Y in the opposite direction so that the X tries to, by natural selection, tries to compensate for that. And that's how I drive evolution of X. So you sort of leverage phenotypic covariation in this case. And eventually what you end up doing, these are sort of the fitness landscapes at different points in time, is you gradually start up from the natural fitness landscape here and gradually move towards a target fitness landscape. So you can do this in a quantitative way, and it's a sort of fun toy problem to work with. But in reality, we don't have the luxury of sort of continuous drive of evolution and also having a very clean model of evolution. So we are most likely closer to this limit that we have basically ability to monitor a population and control it in sort of intermittent phases, maybe in some time period tau, with some time period tau, and implement some discrete control. And what I want to sort of tell you here is based on the theory in a more general way, what are the limits of this control? So one thing we do is if I change this tau and I make it very close to each other, I'm increasing the amount of effort I'm putting in the system. And you can show that the sort of the work associated with the amount of effort you put in is the expectation value of your U square, which was your control protocol. And the power would be 1 over tau if you think about it in a stationary case. So as you decrease tau, you make these control attempts closer to each other. You have a tight monitoring and tight perturbation in your system, and you're putting in too much power for control. The other thing that changes is the amount of predictive information. So every time I intervene with the population, my action would sort of impact the population, but then population starts deviating because of its own natural selection dynamics. And my sort of the impact that I'm making, is basically the memory of that will disappear over time. So as I increase tau, my predictive information decays. So the system forgets what I've done. And this basically introduces a trade-off, how tightly I need to impact the system versus how much information I have. And you can try to sort of draw a diagram as predictive information versus the scale power. And what you end up seeing is that there is basically a bound or information bound for how useful the power you're putting in for control is in a system. And in this context, what I'm showing is as you increase the diversity in your population, so a population tries to evolve faster on its own natural basis, your information bound goes lower, and the amount of power you're putting in becomes less useful. So basically, you can only control the system as much as you know something about the system, right? So the sort of predictive information sets the bound for that. And the sort of the conclusions that come out of this is quite general and doesn't depend on the specific model that you're implementing. So it goes slightly beyond this multivariate phenotype that I introduced. I think I have about 5 minutes or so that I continue,
Yeah,
and I
Armita,
sort
sorry,
of.
I'm interrupting. I mean, you know, in fact, your time is kind of up. So, you know, if you can just be very fast on this last part.
Okay, I'll try to sort of
Thanks.
give you a sense of this new this project that is more related to data, and just we can discuss afterwards. So the project on the control in sort of HIV evolution is a collaboration with the group of Florian Klein in Cologne, and it's led by Colin Lamont. And the idea here is to use broadly neutralizing antibodies, unlike the usual cases that we want to do vaccines with those. We want to use broadly neutralizing antibodies for therapy purposes. And in this case, what you're seeing in the experiment is you have different frequency of the different variants of HIV. And once you inject a bNAb in a person, you see escape mutants showing up in response to the bNAb. And as the bNAbs come sort of decay, because they're proteins, the escape mutants rise, and then they get replaced by the wild type in a population. So this sort of raised the question that there is a cost for escape mutations in HIV against the broadly neutralizing antibodies, and we want to minimize rise of escape against this VNAP. So that's a sort of therapy idea that would replace antiretroviral therapy. And you can pose the same question, for example, for antibiotic resistance or any kind of resistance dynamics that you can think of. To give you a sort of overview, the idea is we want to have a model of evolution that implements cost of escape, the likelihood of escape mutations to emerge in a population, and also have some very minimal model of evolutionary dynamics. And we want to use the possible interventions as a control scenario in this case, which can be antiretroviral therapy and broadly neutralizing antibodies together. And to sort of, I think what I want to basically tell you is we use basically genetic data, like high throughput sequencing genetic data, to build a model for to build a population genetics model for the selection on these escape mutants, and identify target size of escape against different types of antibodies. So this is just statistical data coming out, from basically the work of Richard Ney, looking at high throughput genetic data in HIV. Using those, we measure selection, escape on the escape mutants, and try to infer the distribution of rebound time in different in patients just based on the evolutionary parameters that we infer from the statistics of genomic data. So using a statistics to predict something about dynamics without any extra fitting of evolutionary parameters. And that's useful because the dynamical data that we have for viral rebound in patients are quite noisy, and it's hard to fit anything to those. And so once we did that, we were able to sort of fit very well the rebound time in different trials. So this is one of these antibodies, 10-1074 trial, and these are two of the patients that I'm showing. This is the usual sort of viremia over time, and it's got theory versus data. And so this is a whole distribution of rebound time across, in this case, 16 patients. And so we are sort of able to predict the shape of the distribution for this trial, for a second trial, and another broadly neutralizing antibody with 17 patients. and for combination therapy. So the key here is the sort of the red points, which are the theory predictions. They have no extra fitting parameter that come from this data. So you could give me in principle a new broad neutralizing antibody, and I can tell you the efficacy of this BNAP for how it can be used for therapy purposes. And what the key message out of these sort of theory building here is, if you want to restrict a sort of HIV infection using this BNAP therapies, you need something of the order of 4 to 5 broad neutralizing antibodies that target 4 to 5 epitopes to restrict escape, given the parameters that we have inferred. And so using one BNAP usually leads to escape mutations arising. Alternatively, you can use antiretroviral therapy together with these BNAPs to first reduce the diversity, elongate the growth, the sort of the replication time, or reduce the growth rate of the virus. And by sort of joining these two factors, you can then reduce the number of antibodies that you need to suppress viral escape. So with this, basically, I want to summarize. Well, maybe I'll leave the summary slide for questions, but I want to thank all the people who were involved, basically, especially Colin for the HIV work that I did not get to talk in detail about it, and Jaehoon for the first part of the talk. And this, I'll leave the summary for discussions.
Okay, thank you very much, Armita, for this very exciting talk. So I see that we already have some questions. So Luis Miguel is asking, in the first part, how do the dynamics compare to more classical ones with artificial and natural selection, but no dynamical updating via optimal control? He says that it seems that some level of artificial selection has to persist anyway at equilibrium to counter the influence of natural selection.
Yeah, so I mean, I introduced a specific model of natural selection, right? So it's a multivariate phenotype in a high dimension, so in high dimensions, but in quadratic fitness landscape. And in this case, you sort of the solution for artificial selection would be another sort of quadratic fitness landscape, the strength of which would change over time.
Yes.
And as I showed in the
Sorry, my point, can you hear me?
Yes, we can hear you.
Yes, my point was that there are some models of this kind where you have a multivariate optimum and directional selection. So the multivariate optimum is natural selection, and then there's artificial selection that's directional, which results in a displaced optimum, right? And then, but if you relax artificial selection, so you will reach this equilibrium that depends on the target. If you relax artificial selection, then you go back to the. So my point was that I was wondering whether the results you obtained in this type of framework, where you have an updating of the strength of artificial selection, if I understood well, do you reach a different equilibrium and very different dynamics from those where you just impose a given level of artificial selection beyond the natural selection caused by the multivariate optimum?
So the answer is yes, but then there are multiple things here, right? So we are doing artificial selection that's beyond natural selection. In that sense, we are similar, but we are doing it adaptively. And I don't want to sort of dig into like backup slides because it'll take time, but we can talk offline. What you see in this case, especially if you have a target time to reach, is initially your strength of selection, artificial selection, is very small. And as you get closer to the optimum, you're exponentially increasing the strength of your artificial selection. And the reason for that is that